Meeting is being recorded. I appreciate it. Thank you. But even though we have the exam scheduled for Tuesday, it will not only be, I'm not going to do a synchronous exam. So it won't be during only class time, like 9 to 10:15. It probably won't only be on Tuesday. You'll probably have a couple days to do it. So again, I totally understand crazy schedules. So don't think because it says our exam is Tuesday, March 31st, that it's going to be Tuesday, March 31st from 9 to 10:15. You'll have time to do it. So I'll have more details about that on Thursday. All right. Any questions before we get started on lecture or anything really? All right, we're good? Okay, let's move forward. So here we go. Share screen. Oh, here we go. All right. So what you are looking at here is, let me just set this up really fast. I have a question. So when I have the screen set up like this, are you just seeing my PowerPoint or can you see the chat box as well? Can you just type in the chat if you see both? Awesome, that's what I wanted to know. Okay, and I would like, and okay, and you know, if you set it up, you can also see my face. So you would just go to, I think it's like view, and you can say side-by-side mode for you. So if you want to see my face as well. All right, let's get started. All right. Okay, so we were talking last week. We left off kind of talking about inheritance, and specifically we discussed Punnett squares. So we went through kind of initial Mendel's experiment talking about pea plants. And just a reminder, when we talked about Mendel's experiment and pea plants, that's when we talked about a Punnett square to do a monohybrid cross. So just a reminder, that's what we're looking at on this slide, is a monohybrid cross. To kind of remind you what we're looking at, technically cross, which is what you're looking at with a Punnett square, also occurred kind of like where this line is between P and F1 generation. What Mendel did first is he crossed homozygous dominant pea flowers. So you can see the homozygous dominant for purple flowers, and then he crossed those with homozygous recessive. So remember, homozygous means both alleles are the same. So homozygous dominant means both alleles present are dominant. And then he crossed that with homozygous recessive, which means both alleles are recessive. When he crossed those, we could do a Punnett square right here, but if you do homozygous dominant versus homozygous recessive, all of the offspring will be heterozygous. And remember, heterozygous means that there are two kind of like different alleles present, the dominant and the recessive. So that's what you see, capital P, lowercase p. And then what he did next was he crossed two heterozygotes together, and that's what you're looking at in the Punnett square on the bottom, is the result of a cross between two heterozygote flowers. Sorry. And remember, to set up a monohybrid cross, you just do cross like that. On top of this column, you put one allele, and that would be the first allele here, the dominant allele, capital P. And then the second column, the second allele, lowercase p. So that would be 1 pp. And then along, you would here put the other. So this has sperm and eggs. So write your two gametes. And again, this cross is pp versus pp. And then we just worked that out, and you can see the results of that cross, and you can calculate two different things. So you can calculate the genotype, and in order to look at the genotypic ratio, what you're looking at, how many of the offspring could possibly be homozygous dominant, heterozygous, or homozygous recessive. So if you look at this, a cross between two hets will result in 1 out of 4 being homozygous dominant, 2 out of 4 being heterozygotes, and then 1 out of 4 being homozygous recessive. So that's the genotypic ratio if we're talking about homozygous and dominant or recessive. However, if we look at the phenotypic ratio, remember genotype is the alleles, and then phenotype is the expression of those alleles. And in this case, we're talking about flower color. So remember that in this case, we know that purple is the dominant phenotype and white is the recessive phenotype. So we know that anytime the dominant allele is present, so the capital P is present, we will have the dominant phenotype expressed. So we can look at our ratio and we see that 3 out of the 4 offspring would have a dominant allele present and therefore they would be purple. Only 1 out of the 4 flowers would have no dominant allele present and therefore would have white flowers. So again, that's kind of working through that monohybrid cross. After we looked at a monohybrid cross, we went through a dihybrid cross. So we don't have to go through everything again for this, but I do want to remind you how we set up a dihybrid cross. Honestly, just because like I constantly have to remind myself how to set up a dihybrid cross. So the best way to remember this is to remember FOIL, which is first, inner, outer, last. And that's basically how you set up the alleles on top of the columns. So this cross, this dihybrid cross, we are looking at 2 alleles. We're looking at alleles that code for P shape, which is the R. So either dominant allele, which codes for round, a recessive allele, which codes for wrinkled. And then we're also looking at another allele, so that's why it's dihybrid. We're looking at 2 alleles and therefore 2 traits. So our other allele is looking at P color. So our dominant allele in that case is capital Y for yellow, and then our recessive allele is a lowercase y for green. So we want to know the phenotypic ratio of a dihybrid cross of organisms that are heterozygous for both of these traits. So we'll revisit why in a second. So what we're doing is we're crossing 2 hets. You can see that this organism is a heterozygote for the alleles for P shape and a heterozygote for the alleles for P color. And then we're crossing it with an organism that has the same. So we basically would put this one across the top, this one across the bottom, but you can't just write capital R, lowercase r, lowercase r, because we're doing both of these. So how do you set this up? Like we talked about last week, you use FOIL, or first, outer, inner, last. So for first, that would be the first allele. So for example, capital R, capital Y, and you can see that's the combination we have on top of the first column. Our second column is the inner alleles. So that would be lowercase r, capital Y. You see that's what we have here. Outer, capital R, lowercase y. You can see that's what we have here. And then lastly, last, we have lowercase r, lowercase y. So that's just how you set it up. First, inner, outer, last. I had a question last week that was, could you do first, inner, outer, last instead of first, or first, outer, inner, last instead of first, inner, outer, last? You can do it however you want. You just need to make sure that those combinations are somewhere on top of those columns. So after you get it set up, just like with the monohybrid cross, you start combining these. So capital R, capital Y, capital Y, and then you basically just complete this table. So typically with a dihybrid cross, you could, but you're not necessarily going to care too much about what the genotypic ratio is, but we do care about the phenotypic ratio. So I think this is kind of where we left off last week. So the phenotypic ratio for a dihybrid cross of 2 heterozygote offspring if they follow Mendelian inheritance patterns, like true Mendelian inheritance patterns, which if you remember, so we can scroll back, we have two really important laws during Mendel's hypothesis. One of those is what's called the law of independent assortment. And the law of independent assortment says that alleles for different genes are sorted independently of each other. What that means is if you have two genes, so say for this dihybrid cross we're looking at, for this one, the two genes we're looking at are seed shape, right? So round versus wrinkled. And then the other gene, it's a completely different gene, is seed color, right? Yellow versus green. So those are two separate genes. So Mendel, after all of these experiments, found that all genes are sorted independently of each other. So what that would mean is those genes are not like following each other during crossing over. They're not like associated with each other in any way. They're independent of each other. So if two genes in a dihybrid cross follow true Mendelian inheritance patterns, when you do a dihybrid cross of two heterozygotes, the phenotypic ratio you will get is you will get 9 to 3 to 3 to 1. And you can see where we got that. I think we worked that last week, but basically if we look at our outcome here, 9 out of 16 would have both dominant phenotypes present, right? So that's what we're always looking at, that homozygous dominant phenotype, right? So both dominant phenotypes present. One of the threes is a dominant and a recessive, 3 dominant and recessive. So basically 3 out of the 16 would have the dominant texture round and the recessive color green. And then 3 out of the 16 would have the dominant color and the recessive texture, so yellow and wrinkled. So 9 would have both dominant phenotypes present. 3 would have dominant recessive. 3 would have dominant recessive, basically. And then only 1 of the resulting offspring would, or 1 out of the 16 possibilities, I should say, would have both recessive phenotypes expressed. So that gives us, for a dihybrid cross, if in a dihybrid cross, two genes are not following each other or not associated with each other, we would have the phenotypic ratio of 9 to 3 to 3 to 1. So why do we care? We care because now we know that some genes do follow each other. They are associated with each other on a chromosome. So we'll talk about those later, but I think last week I mentioned, for example, like red hair and freckles. Those are two separate genes, right? So if we were to do a dihybrid cross of red hair and freckles, it would look very similar to this. If they were not associated with each other and they didn't travel with each other, if we did a dihybrid cross for that, we would get a 9 to 3 to 3 to 1 ratio. However, if we really mapped out dihybrid cross of those two genes, what we would find out is that the phenotypic ratio of red hair and freckles is not 9 to 3 to 3 to 1. And that's because they actually do travel together. So we'll talk about those later, but those are called linked genes. So the purpose of knowing that 9 to 3 to 3 to 1 ratio is really to understand that, or basically to understand the how genes are related to each other. Do they follow each other, basically during crossing over? So that's why we care. All right, so let's move on, shall we? I have a couple of clicker questions here, which I know we don't have clickers, and I was going to set up a poll, but even though I had a polling option last week, it's mysteriously gone this week. So I think what I'm going to do is set this up. All right. And I just want to give you a minute to kind of look at it. So this question is, what is the genotype of parent B based off this information. So how do we get this, right? So we're looking at a Punnett square that's already been completed for us. We see the results here. We have parent A in this column, and what we need to know is what's here. Oh, you guys are answering already, good job. So what we're looking at is we know we have a little a coming from here. So what's coming from here? A capital A. We have a little a here, so what must be coming from here? A little a. So that already gave us our genotype. You guys are so good. Right, so we would need a capital A and a lowercase a. So if we have two different alleles, right, capital and lowercase, that tells us it is heterozygous. So like, great job, everybody. All right, perfect. Here's your answer. Okay, so let's move on to the next one. This is in a Mendelian cross for pea plants that are heterozygotes for flower color. What's the probability that the offspring will be homozygous recessive? So before you answer, right, I'm going to give you 30 seconds. You can do a Punnett square, and then when I type go, put your answers in. Okay, so 30 seconds starting now. Wait to type your answers until I say go. It's okay if you're already doing it. We'll do it next time. You're good. Okay, if you haven't already, go ahead and type your answers. You're right, good job. Yeah. All right, hey, perfect. So you can find that out by doing the Punnett square. So I don't know if I did complete the Punnett square here, I didn't. But this is basically like a het versus a het, which is exactly what we did, not there, where am I going? Why does that seem so far away? In this, right? This is also a het versus a het. The only one out of the four would have a homozygous recessive phenotype. Good job. You guys are doing so good. All right, so here's one. So, okay, wait to type your answer. Wait to type your answer until I type go. So here we're looking at a phenotype that is widow's peak. So you can see in the picture, a widow's peak is like when the hairline kind of comes down to a point on your forehead versus a straight hairline, which is where you don't have that point. So actually having that point is a dominant phenotype versus not having it, which is a recessive phenotype. So if you were to cross two heterozygote parents, what would be the chance of having a child with a widow's peak? So if you want to do the Punnett square, that's fine. I'm going to give you 30 seconds, and then I will type go, and then I'll let you type your answers. You have 5 seconds. 2, 1. Forgot I had music there. Awesome, good job, right? So yeah, see, so again, this is a heterozygous cross, just like we did here, except for instead of the question asking about the homozygous recessive phenotype, this is asking about the homozygous, or well, it's asking about the dominant phenotype, but the dominant phenotype will be expressed whether you have homozygous dominant or heterozygote. So that would mean 3 out of the 4 would have that ratio. Awesome, good job. You guys are rocking it. Okay, so we're gonna move on from Punnett squares. are there any questions about Punnett squares or those ratios or anything before we move on? Or is everybody feeling all right? And actually, you know, as always, sorry. I feel like no one told allergy season that this pandemic is happening, so. All right, so let's move on to the fun stuff. All right, so I think genetics is really fun. Punnett squares are not really something I would do in my free time for fun, but I do love talking about kind of like traits and expression patterns. I think it is fascinating. So that's what we're going to do now. Okay, so first, what I want to mention is that up until this point, we've been talking about what are called single gene traits. And again, I'm so tired of having an itchy nose. Again, single gene traits just mean that one gene or one allele codes for one phenotype. So for example, there are several of these that are associated with humans. We've been talking about like flower color or pea shape or pea texture or whatever. On humans, a couple of examples of these are like cleft chin versus non-cleft chin. So you can see in the picture, if you have like an indentation on your chin, that is a cleft chin. That is a trait that is coded for by one gene. Widow's peak, which we talked about in the last example, is also coded for just by one gene. Unattached versus attached earlobes, which you can see in the picture kind of like what an attached earlobe looks like. An unattached earlobe is if you have kind of like the lobe of your ear is separated, isn't directly attached to kind of the skin on your face. That is coded for by one gene. And then also the ability to roll your tongue or not is coded for by just one gene. So those are all what we call single gene traits. And those are inherited and exactly like what we've been talking about in a dominant versus recessive pattern. All right, so let's talk about a couple of examples of, sorry, my parents are here and doing something. All right, so those are all, working from home is so fun. All right, I do love having my parents here though. All right, so those are all examples of what we call single gene traits, where, again, simple dominant recessive pattern. However, well, let's talk about some disorders of that before we move on to like what I like is not single gene traits. So those are what we call Mendelian genes, where it's like one gene. There are some disorders in what are called in those single gene traits. Basically, we can divide those into recessive disorders or dominant disorders. The most common of those are recessive disorders. And what that means is that in order for that disorder to be expressed, the individual would have to be homozygous recessive. So the recessive disorders we know about are like albinism, or severe albinism, cystic fibrosis, PKU, sickle cell disease, Tay-Sachs disease. Those are all examples of recessive disorders. And again, those are disorders where the only time that those are expressed are if the individual is homozygous recessive. So in this case, as we know, because we've done a couple of Punnett squares, the only time that an individual would be born this way is if the parents are both heterozygous, pretty much, for a couple of reasons. So first off, if a parent is heterozygous, meaning they have both the dominant and the recessive allele present, the fact that they have that recessive allele at all makes them what is called a carrier. So they do not outwardly express that disorder. So for example, a heterozygous parent for albinism would not have albinism. They would have the allele for it, But remember, because of the fact that they have the dominant allele present, that masks the effect of the recessive allele. So they would not express that disorder. So they're called carriers. They're carrying the allele, but it's not expressed. That's a het parent. The reason I say, and this is not true for like albinism, the reason I say the majority of individuals born with these disorder have parents that are heterozygous, and meaning that the kind of like recessive allele was inherited from both parents, leading to a homozygous recessive phenotype. Is because for a lot of these, so like cystic fibrosis, PKU, sickle cell disease, Tay-Sachs, and some others, having that homozygous recessive genotype can lead to pretty significant issues that would make it to where the individual would not necessarily reach reproductive age. So that's not all of them. We'll talk about some of those later. But the reason I mentioned like albinism as an example of one where both parents may not necessarily, hold on, if you can just give me 1 second, I apologize. I will be right back. Okay, sorry everybody. Okay, so what they say, like I was saying, so with cystic fibrosis, PKU, sickle cell, and Tay-Sachs, one of the reasons that you wouldn't have like two homozygous recessive parents leading to a homozygous recessive offspring is because a lot of those recessive disorders are so severe that an individual may not reach reproductive age. It's not necessarily true now, but historically. With albinism, that's not necessarily true because obviously that doesn't necessarily impair the ability to reproduce. But a lot of these that are more severe, if a parent is homozygous or if a person is homozygous recessive, they probably wouldn't reach it to reproductive age. So those are recessive disorders. Again, if a parent is heterozygous, meaning they have the dominant allele and the recessive allele, they are called carriers. Because they have the dominant allele present, they don't express the disorder, and only homozygous recessive people express that allele. On the other side, we have what are called dominant disorders, and this is basically like if as you know, if the dominant allele is expressed, that you have the disorder. So that would be homozygous dominant as well as heterozygotes. Because of the fact that in a het, the disease is expressed, you wouldn't call a het of a dominant disorder carrier. So there's no carriers for dominant disorders, just because if the dominant allele is present, the disorder is expressed. These are typically much more severe, and as it says here, typically only heterozygous individuals survive to reach reproductive age. And so with that, that can be a little bit difficult in some of these. So that's not necessarily the case for like Huntington's disease, where people with Huntington's disease is sort of like a dose response. So for example, if somebody is homozygous dominant for Huntington's disease, they tend to get it earlier in life, meaning they would not necessarily survive to reproductive age. But if somebody is a carrier, or not a carrier, sorry, a heterozygote for Huntington's disease, then it's again a dose response. So they actually have it to where they could actually reach reproductive age before they find out that they have Huntington's disease. So the severity for some of these has to do with a dose response of even though the dominant gene may be present, is it homozygous or is it heterozygous? Huntington's disease is an example of that. For some of these, you're going to survive, obviously, like polydactyly. Polydactyly is just present of like extra digits. So for example, like it's very common to have, sorry, my daughter is awake. She'll probably make a visit to one of our lectures, I'm sure. But polydactyly is where somebody may have an extra digit, like an extra finger or an extra toe. And obviously, that is a survivable condition. All right. Some of these, though, achondroplasia and hypercholesterolemia, Huntington's disease is very severe. There are other dominant disorders. The majority of dominant disorders can be pretty severe. All right. I apologize for all the disruptions during that, but are there any questions about single gene disorders? I feel okay? Awesome. Thank you. All right. So let's move on. This slide is crazy, but we will break it down. But this is like one of my favorite slides in all of Gen Bio. Oh, I have a question before we move on. So the question is, what is an autoimmune disorder? So that's a really good question. So, there's a couple of questions. So let's get to these before we move on. What is an autoimmune disorder? An autoimmune disorder is almost always going to be a genetic disorder. There are both recessive and dominant disorders that can lead to autoimmune disease. But autoimmune disease is basically when your body's immune system starts to react against your own body. So typically, your immune system, I guess I should say, in like somebody who does not have an autoimmune disorder, I don't want to say typically, because autoimmune diseases are actually quite common and increasing in prevalence. But I actually have a feeling that this pandemic will eventually lead to even more autoimmune diseases, but that's neither here nor there. Okay, so basically your immune system genetically is built to fight only like things that don't belong. So remember when we talked about like the cell surface and we talked about the sugar pattern on the cells and how we can basically distinguish our cells versus somebody else's cells, or a healthy cell versus an unhealthy cell. Our body is very good at distinguishing even like red blood cells from muscle cells and all of these differences, right? Like your body, your immune system can see those things. Your body can also distinguish your cells from bacterial cells. So this is why you get sick, actually, because of your immune system. Normally this is very good. You get sick because you have a virus or you have a bacteria enter your body, and your body's like, this is not supposed to be here, and it launches an attack against that invader, whether it be a virus or a bacteria or pollen or whatever. It launches a response against that. That can be fever, that can be inflammation, swelling, all of these things. So again, normally your body only does that against things that don't belong. However, with autoimmune disease, which again is typically a genetic condition, your body has some sort of like issue where your immune system thinks it needs to fight something that does belong. So for example, with like rheumatoid arthritis, and we actually don't know why, but with rheumatoid arthritis, your body starts to mount an immune response against something, and we don't know what, but something in the joints. So you get inflammation in the joints. It's actually widespread through the body, but it's most noticeable in the joints. Autoimmune disorders are chronic, and the reason they're chronic is because your immune system will fight that thing as long as that thing is present. So one of the reasons you get sick and get better with an infection is because like you get a bacteria enter your body, You fight it with your immune system, you kill it off, and then your immune system can like resolve. It goes away. It goes away because the bacteria is gone. With autoimmune disorders, they're chronic because you're always fighting yourself, basically. So that's an autoimmune disorder. And autoimmune disorders, some of them are recessive disorders, some of them are dominant disorders. They're tough. And then the other question, are there disorders that only affect one gender, like color blindness? Absolutely, and we are going to talk about those specifically a little bit later. So good question. All right. Yeah. All right. Awesome questions. This stuff is so exciting. Let's talk about it. So here is this crazy slide. But what I have highlighted in yellow, we're going to go through some of these individually. But basically, as the title of the slide suggests, and as we've kind of talked about, up until this point, we've been talking about like single gene traits. So sort of like you have a gene that codes for cleft chin or widow's peak or whatever, purple flowers or white flowers or green seeds. Those are all single gene traits. So they're pretty easy to talk about, and they follow Mendelian inheritance, meaning that if the dominant allele is present, you will have the dominant phenotype. If the recessive allele is present, it will only be expressed if the dominant allele is absent. Like it has these very kind of straightforward expression patterns. But obviously, as we know, we are quite complex, and the majority of genes in the body are not single gene traits, and they have kind of like different and very cool inheritance patterns. So that's what we're talking about on this slide. It's just variations of inheritance. So I have a couple of different examples here as well. So the first type of variation of inheritance I want to talk about is called incomplete dominance. So incomplete dominance is where you have a gene. So for example, on the top of the slide, you're looking at a gene that has two phenotypes, red, well, technically three, as we'll learn in a minute, but what should, if it was Mendelian, have two phenotypes, red color flower versus white color flower. And if this followed traditional Mendelian inheritance patterns, what that would mean is if the dominant allele was present, so capital R, red flower, the flower would be red. If the dominant allele was present in a het, the dominant allele would be expressed, so hets would be red. And if the flower was homozygous recessive, that flower would be white. But as you can see, and this is true in humans as well as several flower plant species, with incomplete dominance, what happens is the heterozygotes don't express fully the dominant gene, and instead the heterozygote phenotype is a blending of the dominant and recessive phenotypes. So you can see here the homozygous dominant is red, the homozygous recessive is white, and the heterozygous is a blending of those two. The het is pink. So that's incomplete dominance. Basically, the dominant doesn't completely override the recessive. Right. So that's incomplete dominance. It's a blending. I mentioned the blending for incomplete dominance because we have another type of variation of inheritance, and that's called codominance. And codominance is pretty cool as well. So that's what you're looking at with these chickens. Codominance is where both alleles are dominant. So for example, here in these chickens, we have like feather color, and you can have an allele for black feather color or an allele for white feather color. And again, whereas like traditionally one of those would be dominant or recessive, with codominance, both of those feather colors are dominant. So what that means is if you get a heterozygote, which is what you're looking at here on the far right, you can see a chicken where both of those traits are expressed equally. So that's a codominance. Both black and white feathers are dominant, and so they're both expressed. So you can see that kind of like striped black and white pattern, which is pretty awesome. All right, so those are incomplete dominance and codominance. We can also have what's called multiple allelism, which I do believe we're going to talk about in a minute, so I'll get to that in a second. Multiple allelism means that there are more than 2 alleles for a certain gene. So for example, a really common example of this is like blood type, because as you know, with blood type, there's type A, type B, and type O, and with those we have combinations. So if you have type A blood, you're homozygous dominant for AA. If you have type B blood, you're homozygous dominant. So it's type B blood would be BB. You can be a heterozygote, which is actually codominance, which you have the alleles A, B. But you can also have O, which we'll talk about that in a second, which is a little bit different. So there's a lot of options, but there's multiple alleles, A, B, and O. So we'll talk about that in a second. You can also have what are called polygenic traits, which is when and there's a lot of these. I would say the majority of traits expressed in humans for sure are polygenic. And we'll talk about why in the next lecture as well. But polygenic, poly means multiple. So what this means is multiple genes influence phenotype. So tons of examples of this, for example, like eye color. I know that I've typically said like, oh, brown eyes versus blue eyes. But in all reality, eyes are very unique. You can see the picture I have here with like tons of different colors. And that's because eye color is actually influenced by a ton of different genes. The genes don't just code for color. They code for a lot of different like, cones and rods and characteristics of those cones and rods, and all of those contribute a little bit to eye color. So lots of genes contribute to eye color. Height, right? If height was a single gene trait and followed Mendelian genetics, we would have tall people and short people, and that's it. Obviously we don't. We have a huge range of heights, which tells us not only is it, the genes contributing, well, we don't actually have a gene that contributes directly to height. We have genes that contribute to like bone growth and muscle growth, and a lot of things can influence it. So skeletal structure, muscular structure, nutrition influences genes in that way. There's a ton of different genes that can influence height. There's a lot of environmental effects that can influence height as well, which we'll talk about in a minute. One very important phenotype that I think is very important to mention as a polygenic trait is autism. So we know very little about autism. But what we do know is that there are several genes that contribute to autism, which is why when it comes to autism, you don't have people who are severely autistic and then people who are mildly or not autistic, right? It's not either or. It is a very broad spectrum of how autism is expressed. And part of that is because it has to do with several genes that are turned on or off and have dose responses. It's affected by a ton of different things. It is genetic primarily though, and influenced by a lot of different genes. That's polygenic. And then we have what's called pleiotropy, which is very interesting, and I'll give a better example of it here in a minute, or a more specific example of it here in a minute. Pleiotropy is when one gene has multiple effects. So we'll talk about this in our next lecture as well, probably Thursday. But one example of this that I will expand upon in a couple of minutes is the gene for sickle cell anemia can actually influence susceptibility to malaria infection. So we'll talk about why in a minute. That's one gene, sickle cell anemia, having effects on two different things. One, sickle cell anemia, obviously, and two, susceptibility to malaria. So that's pleiotropy, one gene, multiple effects. And then we have what's called gene linkage, and gene linkage is what we were kind of talking about when we talked about, and I have the example of like red hair and freckles, probably quite offensive from South Park, so I apologize, but it's a good picture of it. But gene linkage is when genes are linked and travel together during crossing over of meiosis. So I mentioned that the typical example in humans is red hair and freckles. So this matters, again, to revisit our dihybrid cross. If we have a trait that follows typical Mendelian inheritance patterns, meaning that no genes are linked, then we would have that 9:3 to 3:1 ratio for red hair and freckles, meaning they don't travel together. But because of multiple, we know that the phenotypic ratio of a het cross for a dihybrid het cross of individuals with red hair and freckles, we do not get that 9:3 to 3:1 ratio. And this is very observable, as the majority of people with red hair have freckles. Not all, but the majority of people with red hair have freckles. Freckles can be influenced by a couple of different genes, which is why not everybody with freckles has red hair, but red hair typically travels with freckles. So that's gene linkage. And then finally, we can have genes that are affected by the environment. So we'll expand upon this in the next lecture as well, again, probably on Thursday due to time. But some pretty cool examples of genes that can be influenced by the environment are shown here on the bottom left in both Siamese cats and these Himalayan rabbits. I was like, they're bunnies, Easter bunnies. Something that's really cool about these is that these organisms have genes that produce dark pigments. So for example, the cat shown in this picture, if you looked at its genotype would say that that is a fully black cat. Same for those bunnies. But the interesting thing about those genes is that those genes are only switched on in colder conditions. So if you look at the Siamese cat and you look at those Himalayan rabbits, what you'll see is that the black fur gene is on ears and nose and face and paws and tail. So basically on the extremities. Same in the rabbits. Those black fur genes are only turned on the extremities. And those extremities on the body is where the body is most cold. So, which is why you take your temperature under your arm or kind of under your tongue. You're trying to get your temperature in a space that's away from the extremities. More toward, technically rectal temperature is the most accurate, which if you've had a child, you're probably scarred for life because of that. But you try to take body temperature more toward the central part of the body, because the extremities, the ears, the face, nose, paws, hands, tail, those are all colder than normal body temperature. So for those animals, you can see that has effects on color. We will talk about some more environmental effects when we talk about our genetics lecture, and we talk about one of my favorite topics, which is epigenetics. So those would be epigenetic effects. We'll talk about what that means later. So let's expand on a couple of these. So I mentioned the fact that blood type is an example of multiple allelism, meaning that a single gene has more alleles, so more than just two alleles that contribute to their overall phenotypes. So for example, blood type, there are actually 3 alleles. You have A, which is a dominant allele, B, which is a dominant allele, or has a dominant expression pattern, I should say, and then O, which has a recessive expression pattern. So we're not going to go into it a lot, which is unfortunate because it's very fascinating. We may be able to have a lab simulation on it if I can find one, because blood typing is really, really cool with how is it expressed. But basically what these alleles code for is an antigen on the red blood cell surface. So what that means, I'll actually draw a picture of it. So this would be your red blood cell. Let's make it realistic. I'll try to. All right, so here is your red blood cell. If I'm going to do this, sorry, this is taking me a minute. Those alleles code for little proteins. Whoa. I'm going to move this up here so I can move it around. Those alleles, again, you have 2 alleles, no matter what, even though you may have multiple alleles that contribute to a phenotype, you only have 2 chromosomes, each with 1 allele. So this, with multiple allelism, that doesn't mean you have like tons of alleles on your chromosomes. It means that there are more than 2 options for alleles for alleles. So in this case, you have 2 chromosomes, so you have 2 alleles. And in this case, you have like 3 different options. What these alleles are coding for are little proteins on the red blood cell surface, which is kind of what you're looking at here. If you had, I'm going to move this down so I can alter this. If you had homozygous dominant for A, right, so that would be this, your blood type would look like this. You would have both alleles being A. So that would mean you have type A blood. Okay, so let's change this. If, this is a mess, but that's okay. If your genotype was a B, the expression pattern would look like that, and your blood type would be AB, right? So again, those are both dominant phenotypes. That would be your het phenotype. If your genotype was BB, or homozygous dominant for B, both alleles would be B, and that would be blood type B. All right, so those would be, again, codominance because, notice, when we have the genotype of AB, right, you have type AB blood, both of those are expressed, right? So that's codominance. Both of those dominant phenotypes are expressed. So now what about our other possibility? So O is a recessive allele. So let's say you have the blood type AO. This would mean actually that you only have 1 allele. So O actually as an allele doesn't code for one of those proteins on your blood cell surface. So you only have 1 antigen on your cell surface. In this case, AO, that would mean A. So that would be blood type A. Same thing is if you had BO, let's also not, right? You would have B expressed, and you would have type B blood. If, and only if, you had a homozygous recessive genotype, then neither would be expressed, and that is when you have type O blood. So that's a lot, but those would be the genotypes for the different types, blood types. What does the positive and negative blood represent? That's a really good question. So the positive and negative actually represents a whole different gene, which is Rh. So with Rh, which stands for what is called like rhesus factor, and it was named that. I'll move this back in a second. Rhesus factor was named that because it was initially discovered in rhesus monkeys. But basically with Rh, that is a single gene trait. So basically you can have Rh protein, which would make you Rh positive, or you would be Rh negative, which would mean that you don't have it. So if we apply that up here, it's basically the same thing as these antigens. So for example, if you're AB positive, what that would look like is you would have an additional protein called Rh, and that would be AB positive. Does that make sense? So it's just another antigen. That is not, I guess blood type is multiple allelism, but it also is polygenic because there's multiple genes that influence it. So it's multiple allelism based on ABO, and then it's also polygenic because it's dependent on ABO plus Rh. So type O has no antigens. It doesn't have any antigens except for if it has Rh. So if you are O positive, you would have Rh. If you're O negative, there are no antigens. Yeah, at least blood type antigens. So this has a lot to do with the immune system. So one of you guys asked about autoimmune disease. One type of, yes, so what makes O the universal donor? Yes. So, man, I think I took out everything about blood typing and donor and recipient. It had a lot to do with the immune system. Would you guys like me to explain that maybe on Thursday, go into blood typing and donation and how this relates to that? Yes? Okay. All right. I'll do that. On Thursday, what you can anticipate is we're going to move on right now, but on Thursday, we will have a supplemental lecture and we'll talk about blood typing. We'll do that. So I also want to type in here because we just mentioned it. And just a reminder, when we finish this lecture, I will post this lecture with all these notes and these additional pictures and everything as well. Multiple genes, sorry. All right. Perfect. Okay, so I will do that. All right. So let that save. It takes a minute. So blood type, as we found out, is not only an example of multiple allelism, it is also an example of codominance, because with type AB, you have both of those present. I guess I should write that down as well. There we go. And it's polygenic because Rh factor influences as well. So we'll revisit that on Thursday. I do want to continue talking about one thing with blood as well, and that is because effects on blood cells also are a really good example of pleiotropy. So remember, pleiotropy is when one gene would have multiple effects. So we mentioned the fact that sickle cell anemia can affect malaria. So first off, malaria is a parasite that causes anemia, and that's what you're looking at in this picture on the right. You're looking at the parasite, which is shown in yellow, and what happens is the parasite gets into a red blood cell and it replicates to really high numbers, and then as you can see, it kind of like breaks apart the red blood cell. So the reason this results in anemia, which anemia in general is the reduced ability of oxygen to get through the body. So oxygen transport through the body requires red blood cells. So if you have less red blood cells, or if the red blood cells you have are not as effective at getting oxygen through the body, then those can both cause anemia. So in this case, malaria causes anemia because it's like destroying your red blood cells. So not good. However, one thing we know about malaria is that in order for malaria to infect a red blood cell, they have to be like the traditional red blood cell shape. So what you're looking at in this picture, if you look at the very top, right, this circle, that's kind of typical red blood cell shape. They look like cells that kind of like almost like little donuts. They're actually really cute. Like little cells that have been pinched in the middle. They're pinched in the middle because red blood cells actually don't have a nucleus, which is very fascinating. And I think I talk about that later if I haven't already. But red blood cells don't have a nucleus, so they're kind of like donut shaped, flat in the middle. It increases surface area, so they're able to carry more oxygen. Malaria cannot infect a blood cell unless it has this shape. So sickle cell is a recessive genetic disorder. It is a single gene trait, so it follows Mendelian genetics. If an individual is, well, actually, it can have heterozygous effects, but the heads don't express the disorder, or they don't have severe disease, I guess. So it's sort of like a dose response. So with sickle cell disease, what happens is, again, it's a single gene trait. If you have the recessive gene allele present, what happens is your red blood cells form this sort of like sickle shape. So it's called sickle because it's kind of like a sickle, which was that tool used for like harvesting corn, I think. But to me, they kind of look like little half moons. Sickle cell disease results in severe anemia because when your red blood cells have that shape, they have a reduced ability to carry oxygen in the body. However, again, malaria cannot infect cells that don't have their normal shape. So this person on the top, this person that would have normal red blood cells, does not have sickle cell disease, but they are susceptible to malaria because they have normal shaped cells. This person on the very bottom who has sickle cell disease, because their red blood cells are not the typical shape, they cannot get malaria. They won't get malaria. They're resistant to malaria. They still have anemia because they still have sickle cell disease, but at least they can't get malaria. An interesting thing about that is if a person is heterozygous for the sickle cell gene, so carrier, if they're a carrier, even though they have some cells that are sickle shaped, they don't have severe disease. So even though this says they don't have sickle cell disease, they do still have cells that some of their cells are sickle shaped. It's just not, they're not fully anemic. But enough of their cells are sickle shaped that they're immune to malaria. So that's what means pleiotropy is when one gene, so in this case, the gene for sickle cell anemia, has multiple influences. And in this case, it's influencing different types of anemia, either sickle cell or malarial. Super crazy. All right. This is another clicker question. All right. So let's just read through it. I think it's pretty easy. Height can be influenced by many factors. While some genes contribute to this phenotype directly, many do indirectly, such as genes that are involved in skeletal composition, muscle definition, etc. So height is an example of what? What do you think? Good job. You're right. Everybody's right. It's C. It's a polygenic trait, so tons of genes are influencing it. Good job. All right. And hey, we're right. Okay. So I had a question earlier about if there are inheritance patterns or diseases that are more common or more associated with one sex or the other. And the answer is for sure. And these are called sex-linked traits. So, no, those are called sorry, those are called sex-linked disorders, which I think I talk about a little bit later. Or maybe on the slide, where is it? This should be the slide, it's just not outlined well. However, I do want to give this enough time to talk about because there's a couple of these I want to talk about and then we're going to go on, and I don't want to just sort of like jump into this on Thursdays. I think we're actually at a really good place where I want to stop, and this is where we'll pick up on Thursday. So again, on Thursday, what we're going to do, I'll have a little supplemental lecture about blood typing, and we will continue with sex-linked traits. So what that means is we may not get to the genetics lecture. So I may, in that case, extend the deadline for the Learn Smart. Probably not the Learn Smart having to do with nucleotide structure, but the other one having to do with transcription and translation. So I'll have to go through that, make some changes, but of course I will announce those to you. Those changes might not be able to happen until Thursday because I just don't know how far we're going to get. So we're going to play it by ear. But what I can tell you is for sure, based on how we progressed today, probably the vast majority of our exam next week will focus on reproduction and inheritance, and very little will focus on the genetics lecture. So is the mini report still due tonight? Yes. Mini reports are still due tonight for your lab. If you were in my Tuesday lab, your mini report is due tonight, right? So mini reports haven't changed. But we may have some alterations in due dates because I want to make sure we can get through inheritance. This is a really important one for moving on. But then we'll talk about genetics, which is one of my favorite things. Yeah, so due dates will vary depending on basically when you had your lab. Monday or Tuesday is basically should be how it was determined. All right, so any questions before you move? Technically, technically I think we still have like 5 more minutes. So if you have any questions for me, if you don't have questions, let me know. I'll be here for office hours. You know, I can hang around right now, or I'll be able to log on from 11:30 to 12:30 again. I'm just gonna ask, just because of my personal situation is a little nutso right now. If you want to have office hours, like if you plan on joining office hours, just send me a quick email. Like if you would like to meet with me during office hours from 11:30 to 12:30 or outside of that, that's fine. Just send me a quick email so that I just know to log on. But yeah, any questions for me about lecture today or anything? We feeling okay? All right, cool. So I also want to mention one other thing about the exam. Again, I'll talk about this on Thursday. You'll have time for the exam. Next Tuesday, when the exam is scheduled for, which again, you'll have extended time to do it. Next Tuesday, I will still have office hours. So I'll be available for you to like ask me questions via Zoom and everything. So that way, like if you're taking the exam and you have a question, I will be available. So I'll have extended office hours if that's possible. I'm pretty sure I can make that possible, so that while you're taking it, I can be available. And I think that was that. The question, will labs still be due on Sundays? I think moving forward, pretty much labs will be due on Sundays. I just got approval. I mean, just note today, you'll probably get like 1 million notifications from your lab course. And that's because finally today, our lab simulations are being populated into our Canvas courses. This does not mean that all of those are due. So this week, the lab you have due are the inheritance, the 3-part inheritance lab plus the bonus lab, which are posted in our lecture site. They're lab simulations, sort of like we did last week. And those are due Sunday by 11:59 p.m. They're published, so you can start them anytime. Probably moving forward, we're going to open up labs on like Sunday, and you'll have until Sunday to do them. I think that's probably how we will schedule those moving forward, just so that you have a lot of flexibility to do them. Good question. All right. Any other questions? All right. If you do have questions, I'm here to email. What I would like to do is, in the context, we're in cell biology, and because we have this class and we learn cells, I think it's a really good opportunity to kind of discuss what's going on around us. And as a microbiologist, I think it's really important to get good information out there. So what I'm planning on doing in the future is making sure you guys get a good lecture kind of talking about coronavirus. I think it would be good for you to understand what it is. It won't be required for lecture. It'll be completely optional for you to watch just for your own good. I mean, if you're tired of hearing about coronavirus, I totally understand. But for me, something that makes me feel good is having as much accurate information as possible. And this is my thing, so I can give that to you. So I think we're in a good class where we can have that information. So I'm also working on that for you. Yeah, and I also just got assigned to TA, so grading will happen. All right. So thank you so much. Oh, good question. So I have seen that, and I'm pretty sure I'm going to make it my ringtone because I love it. So for reference, though, I have the Kardashians being like, oh God, as my text tone. So that's me. All right. I miss you guys so much. I hope you are having a wonderful, are enjoying pajamas at least. And I will talk to you soon. All right. See you guys later.